Today we will be talking about the microfossils. The fundamentals and introduction of microfossils is something which is required by any student of paleontology for examinations or for starting any fundamental research. Microfossil studies allow precise local, regional and global time stratigraphic correlation that help in hydrocarbon prospect and trend delineation, regional stratigraphic and geological studies and exploitation evaluations. Analysis of microfossils helps us to recognize the paleo-environmental distribution, which in turn is useful to interpret sequence stratigraphy and reconstruct the paleogeography and paleoclimate. Some microfossil function as paleothermometers by undergoing irreversible color changes with post-burial heating. As such, they indicate hydrocarbon maturity levels. Microfossils useful in hydrocarbon exploration can be divided into 5 principal microfossil groups based on the composition of the shell or hard parts. Coming to the composition and the fossil group, like for example, the foraminifera are calcareous, ostracoda are also calcareous, nanofossils are calcareous. Some foraminiferas are agglutinated also. Then radiolarians, diatoms, silicoflagellates, they are all siliceous. Conodonts are phosphatic. Chitinozoans, pollen, spores, acritarchs, dinoflagellates, they are called as paleonomorphs collectively, they are all organic walled. Then comes the calcareous microfossils. Calcareous microfossils have shells composed of calcite or aragonite. These organisms are present in most marine and some non-marine environments. At great oceanic depths characterized by low temperature and high hydrostatic pressure, however, calcareous remains are largely or completely dissolved. The depth below which this occurs, which varies in different oceanographic settings, is termed the carbonate compensation depth. These are 3 principal types of calcareous microfossils: calcareous foraminifera, ostracods and calcareous nanofossils. Coming to the calcareous foraminifera. Calcareous foraminifera are a group of unicellular organisms or protists that secrete a rigid calcite or aragonite shell or test. Fossils of these forms are found in sediments of brackish to marine origin from Silurian to Holocene in age. Most are benthic or bottom dwelling, but a significant group in the late Mesozoic and Cenozoic are planktonic forms. Some stratigraphically important foraminifera developed complex internal structures and frequently large test size. Studied primarily in thin section, these include the Fusulinid, that is Pennsylvanian to Permian, and several groups of so-called larger foraminifera that range from Triassic to Holocene. They occur primarily in carbonate or fine-grained clastic rocks and are excellent time markers. Since many species have limited and well-known environmental ranges, therefore, they are excellent paleobathymetric and paleoenvironmental indicators, especially in younger Phanerozoic rocks. Ostracods are microscopic crustaceans whose fossils are found in Cambrian to Holocene rocks. They occur in most marine and non-marine depositional environments and are generally excellent environmental indicators. The paleontologic application of the ostracods is limited because they are rare in many sections. Many species are endemic to local basins, so their age and environmental ranges are poorly understood. Ostracods typically have rapid evolutionary rates and are useful biostratigraphic tools in some situations. In Paleozoic sequences, in marine environments where wide range species are present, for local stratigraphy in basins of limited extent, in lacustrine environments where they are frequently one of the few microfossils present, ostracods may also indicate thermal maturation of source rocks. Then comes the calcareous nanofossils. The term calcareous nanofossils includes both fossil coccoliths and nanoliths. Coccoliths are minute, minute means less than 25 micrometer. Calcite forms produced by the unicellular marine plants, which is golden brown algae. The origin of nanoliths is uncertain, but these calcite bodies are associated with fossil coccolith assemblages in marine sediments and are also organically derived. Calcareous nanofossils are an excellent biostratigraphic tool because of their rapid evolution and geographical dispersal. That is, their entire life cycle is in the photic zone of the ocean, as well as their varied and distinct morphologies. The oldest known calcareous nanofossils are late Triassic. They are a crucial microfossil groups in calibrating the Jurassic Holocene marine record. Relatively little has been published about the paleogeographic distributions of the calcareous nanofossils. Less is known about the exact paleo-environmental preferences, although they have been shown occasionally to penetrate into shallow marine environments. The main industrial application is their calibration to published timescale and sequence stratigraphic records, especially the association of high abundance with condensed marine sections. Then comes the agglutinated microfossils. Agglutinated or arenaceous microfossils have tests or shells constructed from sedimentary particles which are bound together by organic, calcareous, siliceous or ferruginous cement. There is only one type of stratigraphically significant agglutinated microfossil, that is agglutinated or arenaceous foraminifera. Agglutinated foraminifera are benthic microfossils found in rocks of Cambrian through Holocene age and most marine and brackish environments, particularly in clastic facies. They construct their tests by gluing sedimentary grains together in contrast to the other types of foraminifera which secrete their tests. The sedimentary particles used by these forms may include silt or sand grains, glauconite, sponge spicules or even either foraminiferal tests also. Some species are highly selective in the material used and its arrangement. Although they are sometimes useful in age determination, agglutinated foraminifera are especially valuable as paleo-environmental indicators because they are particularly characterized of the very shallow marine to brackish water environments and very deep marine environments. Agglutinated foraminifera are the dominant and distinctive microfauna in Cretaceous and Tertiary fleshed facies. The color of agglutinated foraminifera has recently been shown to alter irreversibly with heating, both naturally within buried sediments and experimentally also. Now, coming to the siliceous microfossils. Siliceous microfossils are protists. with shells constructed of opaline or amorphous silica. There is no intense dissolution of siliceous remains in the deep ocean. Sediments deposited below the carbonate compensation depth or CCD are commonly enriched in silica by removal of the carbonate, sometimes to the point of forming siliceous oozes. With subsequent remobilization of the silica, deep sea cherts may be formed. Siliceous microfossils are subject to burial diagenesis and become rare at great well depths, except when recrystallized, preserved in nodules or concretions, or replaced by pyrite or calcite. There are three major groups of siliceous microfossils: radiolarians, diatoms, and silicoflagellates. Radiolarians are planktonic protists that occur primarily in open marine deep water settings. They are useful time indicators and are found in rocks of Cambrian to Holocene in age. They may be the only common microfossils in abyssal environments, commonly forming radiolarian oozes. Radiolarian chert, the product of silica diagenesis, is fairly widespread in the geological record. Radiolarians are common in some marine source rocks. Then comes the diatoms. Diatoms are photosynthesizing protists that occur in both marine and non-marine environments. Marine diatoms range from late Jurassic or early Cretaceous to Holocene and are particularly useful for age and environmental determinations in the late Cenozoic. Non-marine diatoms range from Eocene to Holocene and also are useful in the late Cenozoic. These microfossils can be a major rock forming group, forming sedimentary rocks or diatomites consisting primarily of diatoms. Diatomaceous sediments, when altered by burial diagenesis, are converted to siliceous shales or porcelainite and chert. Such rocks can serve as sources and fracture reservoirs for hydrocarbons, for example, Monterey Formation of California. The changes in rock properties associated with silica diagenesis permit seismic definition of silica phase transformation zones in the subsurface, for example, bottom simulating reflector. Then comes the silicoflagellates. Silicoflagellates are another group of planktonic photosynthesizing marine protists. They commonly occur with diatoms. Silicoflagellates range in age from Cretaceous to Holocene. Although not as common as diatoms, they are useful time indicators, particularly in the late Cenozoic. As a group, they were much more abundant during the early and mid Cenozoic than today. They have been used to estimate marine paleotemperatures in the late Tertiary and Quaternary. Then comes the phosphatic microfossils. Phosphatic microfossils, notably conodonts, are composed of crystallites of calcium phosphate or apatite embedded in an organic matrix. There is one type of stratigraphically significant phosphatic microfossils or conodonts, but fish teeth of less practical utility are found in some marine strata. Then comes the conodonts. Conodonts are extinct tooth-like microfossils composed of calcium phosphate whose biological affinities, while poorly understood, lie within the chordates. Conodonts are widely distributed in marine rocks of Cambrian through Triassic age. They are excellent indicators of time and thermal maturity, especially in carbonates, where other methods of evaluating organic thermal maturity are less successful. Conodonts are commonly used as zonal indices for the latest Cambrian through Triassic because they were abundant, evolved rapidly, and were widespread geographically. Although found in most marine rocks, conodonts are most efficiently recovered from the insoluble residues of carbonates dissolved in weak acids or from easily disaggregated shales. Individual conodonts vary greatly in morphology, and taxonomy was originally based on the morphology of these individual specimens. While conodonts are common, the preserved remains of the soft-bodied animal that bore them are extremely rare. Conodonts appear to have been located in the cephalic area and may have functioned as teeth. However, the conodont animal apparently bore in many conodonts of different shapes and morphologies. Based on this study of the very rare vole animal specimens and rare bedding plane groupings of the conodonts representing individual animals. This recent information has led to more accurate multi-elemental species concepts. Let us talk about the organic walled microfossils now. Organic walled microfossils are composed of entirely unmineralized proteinaceous material. There are 4 types of organic walled microfossils, namely chitinozoans 1, spores 2, and pollen, acritarchs 3, and dinoflagellates 4. Coming to the chitinozoans. Chitinozoans are marine organic walled flask-shaped microfossils. They range in size from 50 micrometer to 2 millimeter in size. They occur in rocks of Ordovician to Devonian age. The biological affinities of the chitinozoans are poorly understood, but they may be eggs of marine metazoans. They are excellent biostratigraphic indices and useful paleoenvironmental markers. They also have potential as thermal maturity indices. Then spore and pollen. Spores and pollen are parts of the reproductive cycles of plant and range in age from late Ordovician and Carboniferous respectively to Holocene. Although land-derived, the grains can be carried by wind and water currents into the marine and non-marine, particularly lacustrine and fluviatile environments. The type and relatively abundance of spores and pollen provide useful paleoenvironmental and paleoclimatic information, and they are widely used for basinal and regional stratigraphic correlation. Spores and pollen are also very useful in estimating thermal maturity, especially at temperature levels associated with hydrocarbon generation. Then coming to the acritarchs. Acritarchs are marine microplankton of unknown biological affinity, ranging from Precambrian to Holocene in age. They are excellent biostratigraphical indices for Proterozoic through Devonian strata, but are less important in the Mesozoic and Cenozoic. Acritarchs occur abundantly in fine-grained rocks and are geographically widespread. They have been used for paleoecology, paleogeography, and thermal maturity. Then dinoflagellates. Dinoflagellates are the resting cysts of marine unicellular red algae. They occur abundantly in Upper Triassic to Holocene sediments and are excellent biostratigraphical indices because of their rapid evolution and widespread geographical distribution. Dinoflagellate cysts occur predominantly in marine rocks, but also are present in Cretaceous and Cenozoic lacustrine facies. The morphology and diversity of the dinoflagellate assemblage can be used to differentiate marine environments. We have discussed the basic and fundamental ideas of the microfossils today. We will be discussing about more paleontological groups in the coming lectures to come. Till then, thank you very much.